I'll give you just a little bit of background of who I am and where I work. So I'm a research group leader, which means I lead a group of researchers who do scientific research day to day. And I work at this really wonderful place, which is the Barts Cancer Institute. It's right in central London, near the city, but it's got this lovely green space in front, and we work in this building here. And we, often eating our lunch out here. So, I mean, of course, before the pandemic, and we've been closed down for a large amount of the pandemic, although we're partly open now. So that's a really nice place to work. It's a cancer institute, and it's got about 60 research groups who work across all different types of cancer and all different aspects of cancer. Yeah, it's a very nice, friendly, collaborative, collegiate place to work. And I've been there since 2013 now, which seems like an incredibly long time because it seems to have gone quite fast. This is a picture of some of the members of my lab. I've got a couple of new ones now that aren't on this picture. And this is, of course, the obligatory Zoom. Well, this is our happy hour drinks, actually. We tried to do as much as possible during the pandemic. So these are the important people. These are a group of talented, motivated researchers, and it's my real privilege to work with people like this who share our vision and our interest. And a huge thank you at the beginning. I always thank my funders at the end as well, but I want to then highlight as well, a lot of money, effort goes in, and I'm sure yourselves and like myself too, we all do a lot to fundraise for charity or to donate to charity, and that money is just absolutely what drives us. It's what allows our lab to carry on. And I'm funded by not all of these at once now, but these are now or in the past. So just to highlight that, most of our funding, 90% of our funding, comes from cancer charities. So it makes a huge difference. We wouldn't exist at all without all this amazing support. Okay, so I'll tell you, like I said, it's quite informal, and I've not done too much quite like this before. So I hope you'll find it interesting. What I'm going to try to do is tell you the very basics about what is cancer. I'm sure a lot of you already done, a little bit of reading potentially into this, but this is kind of going to start from the very beginning. Sorry if you've heard it before. But just to set the scene for what I'll say next. And then I'll talk a little bit about these things called chromosomes. Again, you may or may not know what they are. You're just really explaining from the basics again, what are they and why do we care about them? Why do I find them so fascinating? I'll try to inspire you to feel the same way by the end as well. And then lastly, the real link to cancer. So why we actually care so much about chromosomes, and why we've got interested in how cancer cells reshuffle their chromosomes, and you'll see more what I mean about that shortly. And importantly, why we care. Why is this going to be important in the future to improve cancer treatments, or the understanding even of how cancer starts off in the first place? Okay, so what is cancer? So cancer is a disease of cells, and this is a picture here of presumably the top of the skin, I presume here. So there's lots and lots and lots of cells that make up our body. In fact, so many, it's unbelievable. It's actually about 100 million cells, which is a huge, huge number of cells. And there are many different types of cells. So, for example, this is perhaps a picture of, not very accurate picture, but a picture of skin cells on our skin. And then there's very specialized cells throughout our bodies which do particular jobs. For example, we have cells that line our gut, which their job is to absorb nutrients. They have a very different shape, structure, and function. We have red blood cells. They look completely different shape. They have completely different function. And it's the same throughout our body. We have about 200 different types of cells. And most of these do have the potential to form a cancer. Now, something that happens to these cells, we don't, obviously, when you start life, you're a single cell and you become an adult through cell division. Now, cell division is a little bit of a misnomer in a way, because it implies you're taking something and chopping it in half, but really what you're doing is replicating it, you're doubling it and then dividing it into two new ones. So cell division is really cell growth. And as you can appreciate, really, you need this growth to create more cells and more cell mass. And we need that full growth from children into adults. We also need it for healing. If you cut your arm, for example, new cells are needed to come and heal that. And probably one of the major things, especially as a grown adult, that cell division is for, is just replacing old and dead cells. And that is going on at a tremendous rate as well. For example, dead skin cells are coming off from our skin all the time, constantly being replaced by new skin cells at the very bottom layers of our skin. And the same in our gut, for example, as well. Some numbers here, and I still always find this absolutely fascinating. Just in the last minute, an average body will have made 144 million red blood cells, and 35 million gut cells, and 40,000 skin cells, which is incredible. And when, as you see some of the movies I'll show later, you can think about the process that each of those millions of cells has to go through just to copy everything and divide, and it is happening all the time right now, is absolutely incredible to start thinking about. So with all this controlled proper growth, we have a potential problem. So cells have many, many different sets of instructions all contained within our blueprint, our DNA, and I'll come to that more as well later. They have a lot of careful instructions. They tell cells when to start growing and when to stop growing, which is really important. And inevitably this can go wrong, particularly if you get a mistake in the instructions telling you to stop when you've grown enough cells. They can just simply keep dividing. Too many cells are produced and you can form a tumour, for example, here. And that's a really simple overview, but it's one of the key elements here. It's a mistake in a usual control system that's gone wrong. So what goes wrong? How do these instructions go wrong? And how else are cancer cells different from normal cells? So some of the mistakes in our blueprint, our DNA, come from things we know pretty well about these days. Some we don't know so much about. So the things we know more, we know that just as we age, mistakes will creep in. Most of the mistakes are completely harmless, and occasionally through bad luck, a mistake will hit a very important instruction. So that's probably the main reason why cancer is more common as we age, although it's not necessarily completely understood. So just normal cell activities, actually, everything a cell does, lots of things inside a cell actually come and attack DNA, break it down, and that's just a normal part of cell life. The cell, again, has lots of systems to kind of protect against that and overcome it, but just by chance, some of these mistakes will get unnoticed or not protected against. You probably know things about cancer predisposition genes. BRCA1 and BRCA2 are very key examples here. If you have a mutation in BRCA1 or BRCA2 gene, this can be a high risk factor for many cancers, particularly in breast and ovarian cancer, the most well-known ones. So another case, unfortunately, of bad luck really there. And then some things we can do some things about. So for example, lifestyle factors, that's a really, really important one. We're beginning to understand more about that. Smoking, diet, UV light, these kind of things are known, what we call mutagens. They're going to mutate your DNA and give you a much higher chance of getting an unlucky mutation in a very important instruction. And some viruses, and the really amazing example here is human papillomavirus, which causes cervical cancer. And because of that knowledge and understanding, there's now the vaccine, of course, against for young women, which is really, really working. It's an amazing success story. To understand the cause of a subset of cancers and develop a preventative treatment is the absolute pinnacle of what we want to try to achieve. Okay, cancer is very complicated, as I mentioned. It isn't just not stopping that causes cancer. There's a set of 10 elements that people have kind of come to a consensus about over the past several years. And as Vicky said, it is very, very complicated. The literature is vast and complicated. So this set of 10 key elements has actually been a really important cornerstone in the cancer research field. People have used this as a sort of reference to just come back to, and not only to know the important aspects, but for me and other people, for example, we are quite focused in on one area, but it really makes us think about the other things that are also going wrong in cancer at the same time as our thing of interest. And I do think that's really important to think about. So there's a list here. I don't need to go through them all, but the one actually that I'll talk to you more about is unstable or damaged DNA. That's the kind of the root fundamental problem in a way, is that the DNA is saying the wrong thing. And as a result of that, they can grow without being told to, they can multiply forever. Most cells, even if they're in like go, go, go mode, they will eventually exhaust themselves and run out and they just stop. But cancer cells can go on forever. They can do various things as well to do with being in the tissue and hiding from the immune system. And they can evolve, and this is very important because, of course, sadly, most people will actually, if they're unlucky enough to succumb to cancer, it's because they're resistant to therapy. Most people will take therapy, and therapy often works for people. Sometimes it stops working, and that's because the cancer has evolved and a way around it. So actually, this part 2, alongside point 1, are the things I'm interested in, and I'll touch on them really briefly. Okay, so I'll move on to the second part of the talk. I don't know if anybody wanted to ask anything about this sort of very basic introduction of what is cancer before I move on. If I don't hear anything, I'll just keep going. But if you've got any questions, then jot them down. We can talk about it at the end. Okay, so I'm going to tell you now about chromosomes and what are they. So I've put this picture here of this man. This is Walter Fleming from the 1880s. And I love this picture because I always think, now there's a man who spent too long looking down a microscope. with those staring eyes. And, you know, cell biologists like myself do spend hours and hours staring down the microscope. And sometimes you come out of the dark microscope room and you feel like you look just like that. So I love this picture. And this is an important person. He's one of the first people who at least described, drew and wrote down these structures called chromosomes, which he could identify in particular kind of cells. I have to say I've forgotten now which kind of cells he's looking at, where you can really clearly see these without any specialized dyes or equipment. And he noticed these structures. He called them chromosomes because of the, I think it's Latin for chromo, which is stained or color, and somes, which is for bodies. So these colorful bodies he could see. That was it. He was experimenting with different dyes, and that kind of revealed these structures for the first time. And I don't know if I've got them. Yeah, here we go. This is a movie of what happens to these chromosomes. They're not static structures. What they're doing now is moving through a beautiful dance. So this is what happens when you want to grow your cells into 2 new cells. That's what's happening now. So you start off with 1 set of chromosomes, but to have another whole set of chromosomes, you've obviously got to duplicate all of that instruction, make a whole new set of chromosomes, and then accurately pick out from each pair, because they're all different, as you'll come to see, each pair, 1 has to go to this cell and 1 has to go to that cell without fail, without error. So it's this beautiful dance. As I've said, I've got some more movies to show you this process coming up as well. And here's another picture actually of these. This is human chromosomes. There's a method where you can actually take, grow up some cells, take some cells. It's a really fun method. Actually, we do it in our lab as well. You actually create this little test tube of the right kind of solution containing cells, and then you drop it from a height. So we have to stand on a step stool. You drop it from a height down onto a microscope slide, and it sort of splats open the cells and keeps on the slide, stuck to the microscope slide, these chromosomes. These chromosomes are in a very special state. They're in a state they're only in about 1 hour out of every 24. They crumple themselves down into these compact rod-like structures during the process of that separation, that beautiful dance I just showed you. The rest of the time, they're still discrete structures, but they're much looser packed. But nonetheless, there's still a lot of information in here. So again, I'll give you some more numbers coming up here, but it's just to show you that we have 23 pairs of chromosomes. They're numbered 1 to 22, and then we have the X and Y chromosomes. There's no Y chromosome here because this is from a female cell. So we have 2 X chromosomes, whereas a man would have 1 X chromosome and 1 Y chromosome. So that's a bit boring, that picture of chromosomes, and I always love this picture of chromosomes as socks. I think it's a little bit more intuitive and memorable. So here we go. Here's a nice picture of chromosomes as socks for you. As I've mentioned, there's a lot of information here. It's the whole sequence of the human genome is now printed out on volumes and kept at the Wellcome Trust Central Library somewhere, I think in central London. And I like this image because it actually, it's a good analogy for the way chromosomes are. So the genetic blueprint of our cells, this whole thing is a little bit like a set of encyclopedias. Each chromosome is a volume. It's a separate, discrete thing. But all together, you need all of it together. You need all the information. You can't lack 1 chromosome. And then the analogy can continue a bit further, which is quite handy. So each page within your encyclopedia can be a gene, and a gene is a certain set of DNA letters, the DNA code being T, A, C, and G. In certain order, those letters will spell out an instruction to make a protein, which is the actual item in the cell doing a job. So each page might be a gene. And additionally, something that's becoming clearer now, because it's been very difficult to pick out, it's not just genes, it's not just genes on a string that you can read and that's it, it's very simple. It's an incredibly complex process, and organization is really important. And that's a little bit like having, to make a gene into its building block, as we often talk about these things, it's a bit like having a builder and an architect and several project managers. So for example, some project managers might need to be really close to the gene they're in charge of to make it work. And that's often the case. So that means that the exact order of the sequence, not just the presence of the sequence, but the exact order is really, really important in our genome. And that you can tell because we always have the same order. Normal cells in our body have exactly those chromosomes, not, apart from these slight deviation areas between each of us, each individual adult. Apart from that, it's all pretty much identical, and that's really important. I've got a movie here, which was made by a good friend of mine that makes scientific illustrations. This is the stage at which you've replicated all of your chromosomes and you need to separate them into 2 new cells, a bit like the sketch I showed you before. So hopefully it gives you a little zoom in here. each of these, as you can see, have got a neat matching pair. They've been separately coloured, or they're not really separately coloured in the cell, unfortunately. And here we go, they're going to separate now. These kind of wire-like fibres depicted here are real structures in the cells. They're called microtubules. They're tiny little tubule things, and they kind of grow and shrink. And they first of all come and catch chromosomes exactly at the right point. Obviously, it's not going to actually go through, is it? That's sad. I thought I had the movie where it would divide and pull them apart. Okay, so but you've seen the dance I showed you before, and again, we'll have a few more movies coming up. So now going a little bit back in time again, and as you're going to see, we've been working, scientists in general have been working on this problem for a long time, over a century. So these are two important people as well. This is Theodore Boveri, and he worked with Marcella, his co-worker and wife, and I always show her picture because most people tend to only show the man. So they both together noted, looking now at cancer cells down the microscope, and they noticed that cancer cells had defective chromosomes. And importantly, that they failed to separate them correctly into two new cells during mitosis. And mitosis is this process where after you've got your copied chromosomes, you have to separate them properly into new cells. So interestingly, this is a picture here when they'd drawn some pictures, they noticed that the chromosomes here were getting unequally fastened onto and attached between cells. So although this phenomenon was discovered over a century ago, we still actually don't know some essential questions about this, which is mainly why? Why does this happen in cancer? Why do cancer cells like to do this when normal cells don't? So the reason we care about chromosomes and how they go wrong in cancer cell division is back to this issue of the instructions going wrong. So, and as you know as well, cancer is famous for being a disease of mutations. We always talk about mutations. Now, usually, and for several decades, the main focus of cancer mutations has been on the kind of mutation which is a single letter change in the page of our DNA. Now, single letter changes can have big impacts if they're in the right place and in the right gene or the right building block. And they can sometimes completely change the function or completely remove, in fact, a particular machine from the cell. So they can have a big impact, and they are definitely important in cancer. But what in particular our area of research is focused on is in larger scale changes. So we're interested in the changes that happen to whole chromosomes or pieces of chromosomes or rearrangement of chromosomes. And I've shown a picture. These are from diffuse large B-cell lymphoma cells, actually. And you can see this in many different cancer types. It's sorry, I should have mentioned it's very common to nearly all cancer types. Apart from some leukemias. These blobs here are the sort of two nascent new daughter cells, DNA that will eventually become the DNA of two new cells. This blob stuck in the middle is where a chromosome or a piece of chromosome, we can't tell from this picture, is failing that process of mitosis. It's not being pulled away correctly to the other cells. And these are various examples of that. Sometimes strands and strings of DNA and chromosomes also get pulled across. We call these a chromatin bridge. And the consequence of that can be really important. So, for example, if you gain or lose a whole chromosome, you've just gained yourself or lost a whole volume in your encyclopedia, and that's a lot of information that the cell needs. If you chop a chromosome in half and stick it to another one, you've just moved a gene for a particular building block next to the wrong project manager. An example here is really common, actually. It's called a translocation event when you move one piece of chromosome to another part of a chromosome. And there's some very famous ones, the Philadelphia chromosome in leukemia, for example. What it can do, and what that one does, is it can either make a Frankenstein gene, so chopping one half of a gene and sticking it to another to make a whole new Frankenstein protein, or in other cases, it can move a gene that's usually next to a very lazy project manager that only wakes up and makes that gene produce something once in a month or so. And it might move it back to a very active project manager who now says, okay, go, make loads and loads and loads of this gene. And this gene might go on to tell the cell to divide. So the correct order is really, really important of our genome. Also, the number of copies is very important. The cell is not very good at figuring out that it's making too much or too little of something. Again, for reasons we don't quite understand. It's got mechanisms it could use to balance those out, and it doesn't seem to. So that's a quite strange thing about cancer cells as well. So just back to our picture of SOX, I wanted to point out something else here, which is that, and again, very much now getting deeper into our particular research interest, which is that chromosomes are not all equal. Is everything okay? Is everybody following me and happy still? I'm sorry, I can't see your faces or anything at the moment. So
yes, yeah.
Brilliant. Okay, and just do jump in if you've got anything. Okay, so here are our socks. You can probably already see that they've got different sizes. As we get down to number 22, we've got quite tiny little these really annoying ankle socks that fall down if you actually wear them underneath your feet, and really long knee socks here. And this is accurate. This is the depiction of the actual rough length of human chromosomes. They start, 1 is the longest because that's why they're numbered in this order. So they have different lengths, which means they contain more or less information. Actually, the instructions they contain are dispersed very differently. Some chromosomes have fewer important instructions than other ones, and there are many, many other differences between chromosomes, only a small amount, I think, of which we know about at the moment. So they're not all equal, and we think as well that tracking which ones are being affected in cancer is important. And the things that does somebody have a question? No, I'll carry on. The mistakes that Theodore Bavaria and his wife noted a long time ago, we can still see in cancer, as I've shown you. But it's really, really hard still to observe these in a body, in a tumour, in a person, for obvious reasons. We can't go around analysing these things from people. It's really difficult. So what we do, and I've put in a few pictures here, so it's not very polished, but just of what we do in the lab to look at this. So what we do is we grow cancer cells in the lab. We've many people now, and there's many thousands of different what we call cancer cell lines. They're a lineage of cells that were taken initially from a tumour, and they grow in plastic dishes, and we can actually work with them in the lab to investigate what's going on. So this is some of the machines that we use to actually grow these things. And I don't know if you've ever seen anything like this before, but this is our cell culture room. It's a whole special separate lab, essentially, where we do all this work to keep everything decontaminated and separate. Everything is very protected. So the researchers are protected from the cells, and the cells are protected, more importantly, from the researcher. Cancer cells we work with are not dangerous to us. They can't grow in our bodies because they've come from a different human. So our immune system can recognize them. So that's why cancer isn't catching as well. It's only unique to the individual. So we grow in these hoods, this sort of glass here. This glass will lift up a short way, allowing you to get your hands in and actually work. There's all sorts of careful airflow around in there as well to make sure nothing comes in from the outside, because you can easily accidentally be growing bacteria or other contaminations in with your cells. So we have to keep everything extremely sterile and clear and careful. These big boxes here are the incubators. Obviously, cancer cells grow at 37 degrees within our bodies and have a certain amount of nutrients brought to them by the blood system. So we have to mimic that. And this is a picture of inside one of our incubators. We've got some flasks of cancer cells growing in here, as you can see here. We've got lots of different things growing, lots of experiments going on. This is a picture of the side of one of my PhD students, so she's called Sarah as well. She's working in a different kind of flow hood, actually preparing some chemicals which are a bit smelly and we shouldn't be breathing them in. So again, we're highly protected. As you can see, she's correctly wearing her lab coat and gloves, which is very good as well. So we do have to protect ourselves from the work we're doing. And of course, after we've done some experiments, we like to look at the cells, look at the results. And this is our trusty microscope. I've used microscopes like this since 2005, pretty much the same one actually. So I know it really, really well. I've spent many hours looking down this machine. It's so crucial to our work. So here we can track cells. We can do all sorts of things. We can look at cells which are fixed. They've been killed and they're stuck in place, and we can observe them in very fine detail. And we can also take movies, live cell imaging. We take time-lapse photography essentially and paste those together into movies to track cells as they dynamically move through the cell division process. Okay, so I've got a picture here. I don't know if you can see this, but this is again, it's another picture of that key step. Cells have replicated their chromosomes already, and they've moved them apart now into 2 new sets of DNA that will become the DNA for 2 new cells. And I've shown you some examples of this before. These are some chromosomes that have got left behind. And the blue here marks the chromosome. You can't see the cells, but the rest of the cell would be around here. And I've pointed this out. This is a normal cell actually, not a cancer cell. So it shouldn't be doing this. The reason it's doing this is we made it do it. So we, when you're trying to unpick a very complex system, what we sometimes do is start from a much simpler system, for example, a not cancer cell, and we prod it and poke it and treat it and perturb the system to try to mimic what we see in cancer, because that gives us a cleaner system. So we do quite a bit of work with normal cells, and we also do a lot of work with cancer cells as well. So what we did is we prodded it and poked it to make some mistakes. And I don't know if you can notice, but in these green blobs here, they're a very specialized, important piece of every chromosome. I meant to point them out to you before in the socks picture. They're pretty much where the heel section is. That's where those steel cables in one of the movies I showed you actually attach on. So they're a specialized structure to allow them to be grabbed on and pulled apart. And it's a really important structure. And we've colored them all in green here. And in addition, we've colored in all of the of that region in chromosome 1. Now, we all have 2 sets of chromosomes because we have one set from our mother, one set from our father. And in addition, when we're about to divide them, we duplicate those. So we've gone from a pair of 1s to 4 1s. And that's what you can see here. Each red dot here is 1/2 of a new pair of chromosome 1s. And they should be being divided equally to go to the new cells. But interestingly, what we noticed, this particular chromosome, chromosome 1, when we did a particular treatment to induce normal cells to make mistakes, chromosome 1 was really highly affected, and chromosome 2 a little bit as well. And that has started us off on a long journey to understand the differences between human chromosomes and how we can exploit that knowledge to understand cancer a bit better. And I'll come on again to a few more details than this. And I just want to quickly check the time here as well, because I was realizing I was looking at the time on my presenter display, but it's telling me the time and not the amount of time I've been talking for. So that's been confusing me. Okay, so one consequence of making mistakes every time that you divide, and this is a movie now, a live cell movie, is a lot of cells, whole field of cells going through cell division. And this is just to illustrate, in a way, kind of imagine the situation inside a tumor growing, that the cells are dividing like this. And let's assume that every time they divide, they make a mistake. That means that one cell is different to the other. And as you can see, this whole population will go through this error-prone cell division process. And a really, really important aspect of that, and that's become apparent in the last decade or so, is that that provides a huge amount of heterogeneity. And heterogeneity means differences in the population. And it's present in all populations, and it's present in the world. There's lots of heterogeneity in the species. We have different kinds of animals and flora and fauna. It's present between the human population. We're all different from one another, and there's heterogeneity. And there's not just heterogeneity in how we look or behave. There's heterogeneity in how we respond to things, or in how we might respond to disease. Some people might survive the disease, some people might succumb to the disease, for example. And heterogeneity is therefore really important for the survival of a population. If something goes wrong, it's unlikely to kill every member of a population if they're all slightly different to each other. So this is all completely true in terms of cancer as well. And it's probably a big reason why some people can't respond well to a particular therapy. I shouldn't say the people, it's the tumor, of course, we're talking about, but the tumor cells, some of the cells may well respond and die to a therapy, but some might escape because they're a little bit different in a special way. And really importantly, they might escape and regrow the tumor. And that's when we get relapse disease. And that's obviously, again, a huge problem for treating cancer. So we're particularly interested in understanding this process of generating heterogeneity. And this comes back to point 10 on that list before, evolution. So evolution requires heterogeneity. That's how evolution works. Evolution is survival of the fittest. So some things might die off, but the ones that are successful remain, and then they continue to diversify, and then some more pressure may come along, or a new niche might need to be explored. And I really like thinking about evolution as well in terms of chromosome changes. And the reason for that is that there's a quite nice parallel. This is a picture of, if you read the number at the bottom, this is chromosome 1, 2, 3, 4, but the colours are all jumbled up and mixed up. That's because this is a representation of the current human chromosome arrangement, but coloured by where the chromosomes came from in our ancestral species. And again, sorry, this slide was put together a long time, and I can't remember which species this has come from. It's apes, where they have similar DNA, but it's arranged very, very differently. So a lot of jumbling's been going on. This has taken several millions of years. So in massive contrast, cancer cells evolve daily. They make these kind of readjustments, reshuffling changes almost every time they divide, which means that this process here from an ape genome to a human genome took millions of years, but cancer cells can achieve it in days or years, the lifetime of the tumor. And here's a bit of a picture as well. So this is chromosomes, again, colored differently to identify them. These are normal, these are correct. These are the socks that essentially that I showed you before. And if you compare that side by side with the cell from a cancer, I hope you can immediately appreciate there's lots of differences. There's extra copies of some chromosomes. Sorry, the colors are different here, but they're arranged in the same order, 1, 2, 3, etc. You can see that one of chromosome 3 here has been stuck to a large part of what should be chromosome 1. Well, we've already got 2 chromosome 1s. This is an extra bit of chromosome 1, and so on as you go through. Many changes happen. And this is very, very typical. Cancer genomes look like this.
Wow.
It's a huge, yeah, exactly. It's a huge difference. So I'm going to talk just for 5 or 10 minutes, a little bit deeper still into exactly what we do. And then I'll finish up and I'll be really, really excited to hear questions. So what we do, the analogy is like a car engine. So to understand what's going wrong with a car engine, for example, you really need to know how a car engine should work. And that's been what I've done for a lot of my career, is understanding even the processes actually involved in cell division, because it's really complex and we know a lot, but there's still much we don't know. And nowadays in my lab, we're more moving into which parts of those normal processes have gone wrong in cancer cells. So the exact processes, exactly which genes are wrong, exactly which pathways have gone wrong, that leads to chromosome shuffling every time a cell divides. So there are many different ways you can get things wrong with a complex system. If you remove one of many thousands of elements, you're going to get a problem. But there's some hope to figure it out. So some particular processes that we've been discovering do leave their distinctive patterns on the arrangement of chromosomes. And we've also discovered, and others have as well, that some particular processes leave cancer cells vulnerable to specific therapies. So together, this is kind of forming the large overview vision of our research, really, which is to unpick the patterns, to understand the processes that have been driving the patterns, and to then also at the same time discover which processes can we target, which should we be detecting from tumour cells. I'm going to show you another movie because they're beautiful movies. I hope you enjoy. This is a normal cell going through mitosis. We're following the chromosomes. They've been they're marked with fluorescent protein, so we can see them. They're being held by invisible to us, but there are structures there gathering these chromosomes into a central plate. They're attaching to those brighter white dots, as I pointed out before, the section on the chromosome that needs to attach to the apparatus that will move them around. When they're all neatly aligned in a normal cell, they'll do this beautiful movement across. That's called anaphase. That's the part of mitosis when you separate the DNA, and you can sort of see they're forming now new cells. So that is this just wonderful process that goes on. Here's a couple of ovarian cancer cells doing the same process. It's not at the same resolution because that movie was not from our microscope. This is from our microscope. I hope you can appreciate that this cell here is making a big mistake here. It's pulling apart one of its strands of chromosomes. It's just looping around again, leaving a chromatin bridge there, which breaks, as you can see. This next cell, before I start playing it, but you can see that this chromosome here is left out in the cold. It didn't quite make it in yet. So let's see what happens. There we go. The cell does wait, so it's got some things right. It does wait for that one, but it doesn't seem to matter because when it does go through anaphase, it's just brief there, but there's a shot where it's got 2 chromosome bridges going across. So these time lapses we take, it's rapidly sped up. This whole process takes about roughly an hour, and we've sped it up by taking images every few minutes. So cancer cells really vastly mess this up. I've shown you this picture, but this isn't the end of the story. I talked about heterogeneity. So this is one cell, and every cell will look different. And this is a picture of the chromosomes from 4. Again, it's an ovarian cancer cell that we've done this from. 4 cells picked at random. And there, as you can appreciate, again, if you just pick any chromosome and compare it across all 4 cells, there's a lot of differences. And this is just a key example. It's a really good visual example of the heterogeneity we're dealing with in the cells within a tumour. And in our lab, we want to know how we get there, which are the processes, as I talked about before. How can we target this? Again, as I mentioned. And I mean, the thing I really love about our research, although it sometimes feels a bit broad in where we can go with it, I think it's got the potential, because it's such a key element of cancer and how cancer can evolve and become heterogeneous and change its behaviour. We want to understand all the different aspects for cancer, for benefit of cancer patients, which is understanding the causes of cancer, because really prevention is what is going to be really making a big difference to people. Detection is so important. I'm sure you all know that the earlier the detection, the better. And we hope that things we can do as well will help develop better detection methods. And of course, lastly, for treatment as well, as I've already sort of mentioned. So I'm just going to probably skip through a couple of bits here. I'm going to come to this because it comes back to socks. What we've been discovering is that there's particular weak points, just like in your socks, when you put them on in the morning, there's a hole in the toe. It's always the toe, right? That's the weak point. It's the point that you're going to push against the most as well. And it's the same in chromosomes. Particular parts of chromosomes are weaker and fragile than others, and those can get involved in these reshuffling events more often. So we're understanding really when we see a reshuffled cancer genome, although there's lots of differences between cells, there are some recurrent sections which often get broken or moved around, or lost or gained. And we're interested in those regions and understanding why the fragility there and what process was wrong in order to discover these vulnerabilities. And a really nice analogy here, which came from somebody who is in my lab, who's left now, Daniel, he came up with a really nice analogy, which I'll just share with you as well. So learning to recognize these particular defects. So what we might do is we might take an ovarian cancer again, or one of our main models in the lab. We might take a sample from the ovarian cancer and sequence the tumor DNA. So we can understand the sequence. And from that, we actually can build a picture, a pattern of the chromosome structure as well. And from there, this is a kind of pictorial demonstration of the shape of the chromosomes, if you will. So a normal human cell, should have drawn this above, would be a flat line because we have 2 copies of every chromosome. These cancer cells got higher copies of this part of chromosome 1 and lower copies of this part of chromosome 1. And as you go along, there's lots and lots of deviation from what we should expect a flat line. And what we want to know is what painted this picture. What process shaped a normal genome into this, in this particular patient, because this pattern will be different between patients. And the way we can do this is a little bit like recognizing who's painted a particular picture. And you know already, I expect most of you, who painted this picture. But why do you know? Perhaps you've never seen this particular picture before, but you've seen similar pictures by the same painter. What you can look at is the painting style. You can look at the use of specific colors and painting techniques and the objects that have been painted. And these things tell you, even if you never saw the picture before, that it's a Van Gogh in this particular case. And that's exactly what we want to try to do. We want to understand, well, we know when we see particular patterns like this, because of other experiments we've done in the lab, it happens to occur because of a certain process. And then we can start to try to exploit that for therapy. So I'm sorry I talked a bit longer than I meant to there. I hope there's plenty of time for some questions still, and I'm happy to take questions via email and later as well. Another thank you to the lab, and a huge thank you for you for reaching out as well.
That was quite amazing.
Yeah, I didn't just bombard you with too much stuff there.
I know you haven't got long, so has anybody got any questions? I've got some, but has anybody got any questions?
I had one. So I've read an awful lot.
Oh, great. Okay.
And I tried to understand some of the research papers. So it was really good having it pictorially illustrated. Mm-hmm. The amount of changes was just incredible.
It is, yeah.
It's not like just one tiny little, no, that's slightly different. It's like it's all wrong.
Yeah.
Anyway, but they talk about stem cells and fast growing cells as 2 different. How does that fit into what you've just said?
You've actually picked out it's a difficult question. It's an unclear thing. So I mean, I think from your the simple answer is that stem cells, they can unlimited grow. So they have unlimited proliferation growth capacity, but it's more important than that. They have the potential to become any other type of cell, and they're in this kind of early state. Now in cancer, there's a lot of potential parallels to stem cells actually. There's one large body of research that suggests that in the cancer cell population, only some of the cells are actually always growing and keeping the tumor growing. They're called cancer stem cells, but there's a lot of problems because there's a lot of things that don't quite match up with being a stem cell. This genomic instability is one of the major problems with that theory. I think there'll be some answer somewhere in the middle there that reconciles these two, and I've become quite interested in that myself lately.
Yeah. Okay.
But I mean, probably I'll never give you a straight answer to anything, right? Because it's not known.
Isn't there? That's the answer, isn't it? It's a theory then. Rather than
in some cancer, no, everything is again relative to different cancer types. In some cancer types, there's been quite a lot of advances in understanding the very nature of and the appearance of these stem cells, and they really think they can get a handle on these, but in others not at all. So a lot of things will apply to one cancer type and not others.
Yeah, I've heard of the CAR T cell treatment in myeloma, for example.
Yes, exactly. That's great for that kind of cancer, and it's not going to be applicable to all cancer types.
Yeah.
Yeah.
I had a question, it's probably stupid, but I'll ask you anyway.
No stupid questions. I meant to say that at the start. There's not any stupid questions.
Can DNA be altered to redirect instructions to cells? I know that there was a couple of ladies that won the Nobel Peace Prize because they've worked out how to change DNA. Will that be something for future reference?
Possibly not. So in cancer, I mean, for disease, yes, it has the potential. When a disease is caused by a single letter change, for example, muscular dystrophy, other genetic diseases, yes, because you can maybe get to the right cell and fix the error. But I think you can probably appreciate that you're not going to fix a cancer cell with a little bit of an edit. So yeah. You might be able to make it do something else, but it's always this heterogeneity. It's always coming back to that. You might fix it in some cells, but not all.
Okay. I suppose you just really want to get it to remember to die.
That's true. We might die. So if you can reintroduce correct instructions where they've been overridden, possibly. Again, only if you know exactly how the cancer cell got around it in the first place. And the other problem, right, is cells are so complex that there's so many pathways around a block. That's why they're so resilient, is that there's so many ways to get around a block.
It's amazing. With the translocation events, does that happen in most cancers, or is it just certain types of cancers?
There will be translocations in most cancers. Now, are they important for the disease is a very different question. So the one I mentioned in leukemias, for example, leukemias will often have not much going on actually, but one important translocation. And that's been selected over time. We see it over and over again in different patients because it just happened to put, like I mentioned, two genes next to each other that then can drive the cancer. So some tumors are driven by a particular translocation. Many others will have them, but we don't know their significance.
Interesting. So I think it's fascinating. I mean, I can see why you spend all day every day doing that.
Yeah.
It's quite amazing. I was just amazed by the amount of change, I think, is what I've taken away from that in terms of how it's affected the chromosomes and the change of the socks.
Yes.
That's amazing. I mean, I know that you need to sort of get off and do other things this afternoon.
But I've got till just after 3, so I'm really, I didn't mean to talk so long. So
I mean, I don't know if anybody else has got any questions, any burning questions about the genetics of cancer or the sort of science behind it. I mean, it's so complicated. Trying to understand it is, well, for a normal person, it's quite complicated.
I am a normal person, by the way.
You are normal.
It's complicated for me as well.
But, I mean, it's quite fascinating. But I mean, I'd just like to say thank you for your time this afternoon. I think it's been really interesting. And I know there's a couple of people that would love to see the recording as well. So I will share that with them. If there's any questions that come out of that, I'll drop an email to you if anyone has any burning desire to ask about chromosomes or socks or anything else.
Yeah, sure. That'd be great. I mean, as I mentioned as well, I really appreciate that you reached out. I think most people, I know people are busy, but most people are very, very happy to try to talk about what we do. And I mean, as well as I sort of mentioned, I don't know if it might be something that you could help me out with in the future, is that very often when we apply for money, it's more and more people would like us to be in contact with patient support groups or networks or patient advocates to get the feedback really on, do you think our research is even useful or important? It's really hard for us to get in contact in a sort of formal way with people. We can't just say, oh yeah, my aunt says what I'm doing is really important. It doesn't work. So I probably would be in touch in the future if you don't mind.
Well, that's fine. That's absolutely fine. Yeah. I mean, it goes both ways. It's fascinating for us to find out about this type of thing. I mean, because we want to understand the disease as well, as well as obviously we'd be more than happy to help out with any sort of testimonials or any feedback or anything like that, really.
Thank you. I mean,
what you do is vital, so Yeah, we'll do everything.
Thank you. No, like I said, yeah, hopefully, I mean, I'm trying not to write any more applications at the moment because it takes up a lot of time, and I have some funding at the moment. So, but in the future, I wouldn't be surprised if we, you know, if
we can, certainly help with that. I mean, you know, we've got lots of contacts as well that I'm sure would be more than willing to help on that front. But no, it's brilliant. So I would just like to say thank you, really, and I know everybody feels the same. I think we're just appreciative of your time, and I know that you're very busy. You probably spend a long time in that lab.
Well, not as much as I like at the moment. I do manage to get in a couple of days a week now, but a lot of it's remote now, which is not the people aspect is it's really important in science, you know, to chat, brainstorm, and you know, we're lacking that at the moment. We're trying to get back to it.
Yeah, it's a funny world we're in at the moment, isn't it?
Isn't it? Yeah, well, I wish you all the very best of luck with all of your journeys onwards and treatments and everything, and it's lovely to meet you, and hopefully we can stay in touch then in the future.
Yes, definitely. No, we'd love to stay in touch. But we'd like to say thank you very much.
Thank you. I'll run off now and pick up the little rascals. So.
Yeah.
Thank you.
Bye.